Okay, so I'm going to jump right into my research for the first part of this talk. Last summer, every week, me and two colleagues would go out to the Richmond Wildlife Management Area. We would bushwhack through chokecherry trees with tent caterpillars on them, walk through a patch of glacier lilies and a dry riverbed, and arrive at this open green meadow with research plots marked off on them. We would get these plant sampling quadrats, put them down, get on our hands and knees, sift through the vegetation, looking for and counting the number of this noxious weed leafy spurge to estimate its abundance there. Now, after an afternoon of doing this, counting about 80 quadrats worth, we would have seen dozens of different grasses, the occasional purple silvery lupine forb, but only about 2 leafy spurge stems per square meter. Now, data from about 20 years ago suggests that there was about 370 stems per square meter in the same area. Now, why is there such a radical difference in abundance in this weed between now and then? Because ecologists have introduced this host-specific flea beetle from Europe, where the weed itself is native. These flea beetles will lay their eggs in the soil, and the larva will eat the root tissue of the plant, limiting its reproductive capability. Now, that's my research in a nutshell, the long-term effects of this type of weed control. Now, why am I interested enough in this plant-insect interaction to approach a nice mentor and spend lots of hours looking into this, studying it, and writing about it? It's because I'm attracted to details, specifically plant details. I like to pick up a plant and look at it for its identifying features, where it tends to be found in a given habitat, its history, its cultural uses, native or non-native, and if it's been used for forage or medicine or food. And 2 years ago, some friends and I decided to test how much we thought we knew about these details. So we went camping for 4 days in Ogden Canyon, bringing only plant field guides, sleeping bags, and lighters to see if we could survive. And we did. We weren't full, but we did survive. And we had delicious meals like burdock dandelion salad with insects for protein and rosehip yarrow tea. It kept us going, you know? And after that experience, I sort of developed an awareness that details give you. I looked at nature with new eyes. What was once just a landscape and a field of grass to me became dozens of different species of grasses all interacting together in a community, and they all each have their own stories. Now, this sort of process of starting at details and working your way up is very applicable to research as well. For my project, I had to start at the roots of this weed and this type of weed control. So I saddled up to the computer and I started studying the biology of the weed, the biology of the insect, the theory behind biological control of weeds, studies that have been done in the past with this weed and insect in North America. And after spending hours looking into the biology of these things, I began to formulate a story in my mind about what's going on with this insectant plant. But more importantly, since it's research, me and my advisor looked for keen missing chapters of this story, details that are still largely unknown. And that's what we got to look into, such as, after a couple decades of being controlled, what vegetation tends to replace this noxious weed over time. And what we found was that all forbs and all grasses that were at the site previously increased significantly in abundance, and that specifically it's introduced grasses that now dominate the site, a finding that can be helpful for farmers, land managers, conservation biologists. And that's one major detail we got to fill in. Another detail of the story was which of the 3 flea beetles that were introduced tended to survive in a dry habitat like Richmond over time. And we found that the black flea beetle is around in the most abundance relatively now, while the brown flea beetles have decreased. Now, spending weeks out in this meadow, we also observed lots of things we didn't expect to see, such as the cerambycid longhorn beetle and its potential negative effects on leafy spurge flowering capabilities, as well as potential different population dynamics in a shaded, wooded nearby area compared to the open, dry meadow. These are new chapters of the story that are interesting, but we didn't get an answer with such clarity. Now, after spending a year and a half roughly on this project, I've developed a sort of reverence for this research process of starting at the roots and working your way up, getting to know the details, formulating a story in your mind, getting to answer some missing chapters, and developing new questions that can be answered in the future, and ultimately arriving at a new state of awareness. And that's what I'll end with. So I can now look at the field of science with new eyes, knowing that the macro big picture and the micro details are intimately connected, and one cannot be completely understood or appreciated without the other, and how everything we think we know about the world came from a series of persistent observations. Thank you.